Hi. Meeting in progress. Got it. Okay, well, thank you all for coming inside on this unseasonably beautiful day here in Moss Landing. And I think it might be unseasonably beautiful up in the Bay Area too, which is where our speaker is coming to us from virtually today. Happy to welcome you to the Moss Landing seminar and very happy to welcome our speaker, Dr. Michelle Jungbluth, who is coming to us from the San Francisco State University, the Estuary and Ocean Science Center, where she's been for a few years in her capacity as adjunct assistant professor of biology. And prior to that, she did a postdoc at SFSU in the Estuary and Ocean Science Center, studying copepods and all sorts of very interesting food web interactions and the systems there. And hopefully she'll tell us a lot more about that today. And prior to that, she did her PhD and master's in oceanography at the University of Hawaii at Manoa. So she is familiar with copepods and all sorts of trophic interactions in a variety of different ecosystems. And with that, I will just like to say welcome, Dr. Jungbluth, and we're excited to hear from you today. Are we good?
Yes, thank you.
Okay, great.
Can you hear me all right?
Yep.
All right, fabulous. Thank you for that introduction. Yes, I'm excited to be here today. Thanks everyone for coming to my talk. I'm very excited to present on some old work and some new work looking at the hidden diversity, abundance, and feeding interactions at the base of aquatic food webs. So just a brief outline. My research in general tends to cover three main themes. Those are marine and estuarine food web ecology, larval zooplankton ecology, and plankton diversity. In my talk today, I will cover the why and how I study all the small things, from zooplankton to fishes and their roles in marine food webs. And then I'll go into some background about my early research in Hawaii doing eDNA before it was cool, that's environmental DNA. And some DNA barcoding, quantitative PCR, larval copepod studies that I did there. And then I'll fast forward to my work more recently in the San Francisco estuary, looking at what provides food web support for zooplankton in the San Francisco estuary, and look at zooplankton diversity and larval fish diets in the San Francisco estuary, and then very briefly talk about new and future work. So just to get us all on the same page, I'm sure most of you know what plankton are, but for those that may not, plankton are organisms that float along at the mercy of the tides and currents. In Greek, plankton means drifter or wanderer. There are three general categories of plankton that we like to classify them into broadly. The bacterioplankton, which are the ultra microscopic bacterial portion of the plankton. There are the phytoplankton, which are the photosynthetic ones that are the primary producers at the very base of the food web. And then there are zooplankton, the animal portion of the plankton, which tend to be the focus of a lot of the work that I do. And when I first started in the lab at the University of Hawaii, I was actually a volunteer in Erica Gertz's lab. She does copepod population biogeography. And when I started there, I started volunteering, and it was a DNA barcoding project, identifying copepods from mixed samples and then isolating them for characterizing DNA sequences from each of them. And I instantly fell in love with plankton. Before that, I knew nothing about plankton. I was a terrestrial biologist 100% before moving to Hawaii. But I fell in love with them partially because you can see there's just a variety of body forms and colors and transparency and all these fascinating features that they have that they've adapted to the aquatic environment. And it just makes me fall in love with them every time I see them. So that's pretty cool. Not only are they cool and diverse and they're also critical members of marine food webs. Zooplankton fall at the space between the primary producers and the bacteria that remineralize everything. And their zooplankton are critical food resources for pelagic planktivorous predators, specifically things like larval fishes rely heavily on zooplankton prey to get through those early stages of fish development to become bigger and stronger, and the larger fishes that we tend to know more about. And within the zooplankton, much of my work to date has focused on marine planktonic copepods. Why, you might ask? Well, I like to use this photo to illustrate why. This is a picture of what you might see if you towed a net in offshore Hawaii, which is an oligotrophic subtropical region. And I did this around Kaneohe Bay. To the untrained eye, you might look at this and think that it looks like a bunch of unimportant gunk, maybe a couple things that look like bugs, maybe some things that might be larval fishes. When you look closer, I've gone and circled everything that I could identify to be different species or life stage of a copepod. And I like it because it sort of illustrates that they are, the facts that they tend to be the most abundant type of zooplankton in marine and a lot of aquatic environments. And they're also highly diverse. And so that means they are copepods themselves are very important members of marine food webs. Now, we know a few things in general about plankton diversity that sort of drive bigger questions about how plankton may change in response to climate change. One broad pattern about plankton diversity is that in the tropics and subtropics, you have a much higher species richness, or a number of different species in the sort of warmer latitudes. And as you move towards the poles, the species richness tends to decrease. And that is generally a result of temperature and resource stability. Near the tropics, of course, you have more consistent light patterns, and even though generally low, the nutrients tend to be relatively stable. And so that tends to support a higher diversity of many different species. Unfortunately, we know that as a result of warming and climate change, zooplankton compositions are shifting globally. And for the most part, we're starting to notice warm water species starting to travel northward, and even the cooler water species traveling poleward. And that raises some questions about how is that going to impact food webs? And we know from prior work, even things in as early as 2003, that it is already, there are clear connections between the climate oscillations in the ocean, like the Pacific Decadal Oscillation, which is between warm and cool periods, things like impacts of the blob that happened a few years ago, and the assemblages of copepods in certain regions that overlap with things like coho salmon. And you have changes in coho salmon survival that are related to the assemblages of copepods that are present, specifically the northern copepods, which tend to be more lipid rich and more resource rich, providing more resources per unit consumption. So that raises some huge questions about how are these types of shifts going to impact other marine food webs or other aquatic food webs. But that's a pretty big question to answer, especially when we're still learning so much about the diversity of species. And sometimes to answer the big questions, you have to start small. And that's why a lot of the tools that I tend to use in my work uses DNA as a tool to study food web connections and ecology. And DNA is one of our most powerful tools in the age of biology. Every living thing has DNA, which codes for proteins that build life as we know it. And before I get into my actual research, I'll just briefly introduce some of the common DNA techniques that I use and that I'll refer to. One of them is DNA barcoding, and this tends to be targeting single organisms, where you're targeting a single gene within that organism and get a DNA-based ID or a DNA-based DNA sequence for that one organism. With community DNA metabarcoding, that is sequencing a single gene region with general primers targeting everything in a sample with whole organisms using high throughput sequencing. So that generally sequences all the things in whatever sample you're looking at, but it's like a water or a zooplankton sample sequencing the organisms. There's also dietary DNA metabark coding, which is similar to the above, but it's on guts or feces usually. And then there's environmental DNA. Now that can be confused with community DNA metabark coding, so that's why I'm kind of trying to specify here. Environmental DNA is generally used to characterize free-floating DNA or loose cells that are not locked up in a whole organism. So this is usually either one of two things. You're doing a single target species using species-specific quantitative PCR, looking at detection or sometimes quantification of one species of interest, or characterizing a whole community environmental DNA, the free DNA, using general primers that target everything using high throughput sequencing. Hopefully that's makes sense. So I'll refer to these terms throughout my talk, so that'll help it be a little more clear. So not only is DNA ultra small, but so are the organisms that I began my research studying. And those are copepod nauplii, the earliest life stages of copepods. Copepods, after hatching from the egg, develop through 12 stages. They hatch from the egg into a nauplius, develop through 6 nauplier stages, and metamorphose into a peptidite, which is more reminiscent of an adult copepod. They molt through 5 nauplier stages and then molt into the final adult stage. So it's kind of complicated, but over the course of their development, they increase in size by roughly an order of magnitude or more, depending on species. Now, because they're so small, that makes them very important in food webs because they also tend to be much more abundant than the adults. Many of them die off before they reach the adult stage because they're important prey. And we know that larval fishes rely sometimes heavily on copepod nauplii, especially in their earliest feeding stages. Unfortunately, they're very hard to identify. And I use these two pictures as an illustration. These two species, Bestiolina similis and Parvocalanus crassirostris, they coexist in tropical and subtropical waters and around the world. And as adults, you can tell them apart, though with some challenge, but as nauplii, it's basically impossible to morphologically tell them apart. And we know that even though they may look the same and they're the same size, they do start to have species-specific behaviors in their early development that may make it very important, may mean that there are important differences between species on how susceptible to predation they are and which fish may be able to prey on which species. We know by fish larvae, there is selective feeding on adult copepods, but because they're so hard to identify, it's often hard to determine whether there's selective specific feeding on different nauplii species. So with my master's work, I set off on creating DNA-based tools for studying nauplii. And so I had the question of whether we could use a DNA-based method to estimate the abundance or biomass of nauplii in mixed field samples. And we had a hint that this would probably work from prior work by Durbin et al. 2008 that was able to describe the relationship between body carbon or biomass and mitochondrial DNA copy number using quantitative PCR for Acarcia tansa in culture. And they developed it so that they could look at predation on the species, but they did find a nice relationship between biomass and DNA copy number. So I set off to do this for one of the species in my study region, which I'll detail next. But first I needed to characterize what species were there and figure out their DNA sequences. So the field site for my work was Kaneohe Bay, Hawaii, on the east side of Oahu. In the southern part of Kaneohe Bay, zooming in here, it's relatively well described from prior studies because of its closeness to the Hawaii Institute of Marine Biology, a marine lab that's part of UH Manoa. In this region and in surrounding waters, it tends to be nitrogen limited with relatively low nitrate and phosphate. The chlorophyll A tends to remain low except during environmental perturbations like storm events. And it tends to be dominated by the small cells of cyanobacteria synechococcus. So in my first task was to characterize the diversity of species in Canyon Bay and get their DNA sequences. And I set out to collect samples in the South, Central, and North Bay. And I found three dominant species. Well, they weren't dominant, but they were found regularly up in the northern part of the bay that were really only found far north. Whereas there was one additional species found from the mid-bay northward, and 4 that were present from the South Bay through the North Bay and were the most abundant throughout Canyon Bay. So those ended up being the main 4 species that I targeted. I characterized the diversity of species present using DNA barcode, the mitochondrial CO1 gene. That is a good gene for being species specific because of its low rate of mutation within species. So now that I had the DNA sequences for all of the species that I might come across around my study region, I set off on designing a quantitative PCR assay to estimate species abundance. And this is what I like to say is environmental DNA before it was cool. So that aspect of environmental DNA that uses species-specific primers for detection and sometimes quantification of your species of interest. So in this case, I was able to use those sequences to design primers that would amplify only one species out of a mixed sample. And after a range of testing and validation, I came up with a successful primer set. And from cultured samples, my first species was Parvocalanus crassirostris, and it was one that's common throughout the estuary, one of those ones that was south to north. And we were able to culture the species, so that made it easier to make measurements on individual naupliar and copepodite stages in the adults. So we found that there was a general increase in DNA copy number per individual. And you might notice that it increases by quite a bit between the naupliar and adult stages. So I also developed a size fractionation-based method to separate the large animals from the smallest so that I could get a better accuracy when working with mixed field samples of many mixed life stages. In the general workflow, oh, that didn't show up very well. Oh, that's a first. Okay, well, anyway, was to use that calibration I showed on the prior page for the qPCR measurement of DNA content. Oh gosh, that's totally wrong. Well, anyway, take a mixed environment. The idea is ultimately working with field samples was to use mixed environmental sample to pour through the size fractionation tower to separate large things from the smallest nuclei, preserve and extract each size fraction, extract the DNA, and then use qPCR to measure the DNA content in a size fraction. Then divide that by the average individual in the size fraction to estimate the number of DNA copies per, gosh, I can't believe that that is not showing up at all. Well, anyway, to estimate the amount of individuals that were in the mixed field sample based on these calibrations. Sorry about that. I hope that doesn't happen more. So doing that workflow, I found that there was a very nice one-to-one relationship between the count in split paired samples between the counted number of nuclei in a split paired sample to the qPCR estimated number of nuclei. So it seemed to work really well to estimate the abundance of these species. And a few years later, I was really excited to find not my first citation, but one of the most exciting citations of that work, actually used my method, developed an assay for the crown of thorns sea star, which is it is an invasive and very damaging species that just eats the reef, basically, to look at the distribution of pelagic larvae in the water column, and they were able to use it to track the abundances of the larvae and where the peak abundances of this species are. And I think it was temporal and spatial. So it was just very exciting to see that my work was sort of being cited and used in a way that really informs us, because otherwise it's really hard to know what larvae you're looking at in general. And more recently, I have a paper currently in press where we actually designed 3 additional assays for those the other 3 dominant species in the South Bay, and characterized the biomass to copy number relationship, and found that there was a robust relationship between DNA copies and biomass of these species over 5 orders of magnitude. From single animals, it gets a little fuzzy, but up to mixed samples, it worked very nicely. So the next steps are that my last chapter of my PhD, which I'm still working on publishing, was follow-up work that I did chasing storm events in Kaneohe Bay to look at the response of the copepod population through this qPCR lens, characterizing the biomass and abundance of Malpii. So stay tuned for that in the next year or 2. Fast forward to my work in the San Francisco estuary. So after graduating with my PhD from Michigan, I moved to the Bay Area to start a postdoc with Wim Kimmerer at SFSU, studying food web support for fishes in the San Francisco estuary. Before I talk about my work with fishes, I'm going to stay in the zooplankton realm, but the zooplankton are still relevant because fishes are declining, and we think that it's partially a food problem. And we think it's partially a food problem because they're declining because the San Francisco estuary, it's a complex system. There are a number of complex interacting factors that are contributing to the decline in fishes. One major cause is that in the zooplankton and benthic assemblages, there have been a number of introduced species that were mainly a result of ship ballast water from international waters. And one of the major introduced species was Potamacorbula amurensis, shown here. And it was found that shortly after its introduction in the mid-80s, it so effectively filtered the phytoplankton from the water column that chlorophyll has never been the same. It has drastically reduced chlorophyll over a range of salinities throughout the estuary since then. And so that has decreased food web resources from the bottom up, and potentially that is one cause for the decline in fishes. There's also other factors like changes in freshwater patterns and use around the estuary, which when we have less fresh water coming into the estuary from droughts, that decreases spawning habitat for anadromous species and native freshwater fishes, and decreases supporting these critical habitats for these species. Focusing in on the chlorophyll story, chlorophyll, because it's an indicator of primary producers in phytoplankton, it's commonly used as an indicator of food for zooplankton, or these food resources that are available. But we're starting to see that chlorophyll is not always a good indicator of food for our zooplankton in the estuary. This is a recent study by Geerty et al. 2021, where it was sort of a meta-analysis of work in the Kimmerer lab over the years, looking at chlorophyll levels as compared to the egg production and growth rates that were measured for one of the dominant copepods, Pseudodiaphanous forbicide. And they found that growth and egg production rates are only very weakly related to chlorophyll. I don't know if I would call that a relationship. It's very weak. There's not a clear relationship between them. In additional work that I participated in, we looked at the I helped design a qPCR assay to look at ingestion of a phytoplankton bloom, the dominant organism, which is Elacosyra granulata, and found that the ingestion rates of the primary organism was often extremely low, and it was totally unrelated to egg production. And there was no relationship between chlorophyll and egg production or growth in that study either. And it's not a huge surprise. We know in other estuaries, this may be the case as well for some species. For example, Acacia in Chesapeake Bay, for that species of copepod, egg production is more related to protist biomass, not phytoplankton, and it's related to temperature in Chesapeake Bay. So it may just be the case that in some estuaries, chlorophyll is not providing the main food resource, and that's what it is. It's hard to say if that is the case before or after the introduction of with Potamacorbula, but we know that that's the case now. So something that we've been pursuing the last few years is trying to define what really defines food for our copepods. And traditionally, the methods usually involve either gut dissection, which can't really be done for copepods, can only be done for larger species, larval fishes, it's even tricky, but yeah, for copepods, you can't really do that. For most zooplankton and copepods, you have to do bottle incubations if you're going to do it traditionally. And that requires comparing assemblages in water before and after incubation, using control bottles with no predators to look at natural rates of growth versus the treatments that contain predators and look at what's removed. Now that's trying to interpret that tends to be not so straightforward. Sometimes there are trophic cascades and there are a lot of complex interactions happening. So the field is starting to move towards more advanced methods, or at least pairing those with traditional incubations. One of those is gut fluorescence, but that only tells you about chlorophyll-bearing cells, the phytoplankton ingestion. And then there's DNA, doing things like the targeted quantitative PCR assays that I mentioned, and doing high-throughput sequencing and looking at dietary DNA, which is tends to both be my specialty. And we know that doing dietary DNA from a range of other studies, you get many previously unknown prey, from studies on very small things to even larger things. And that's because DNA will identify even the soft-bodied things that are hard to identify as species. And many of the things that I've sort of put in bold are the soft-bodied or hard to ID. So we knew going into these studies that doing DNA dietary analysis would get us a lot more information, which is great. There was one prior dietary DNA study on our dominant copepod, Pseudodioptimus forbizae, by Ann Holmes, who was a Kimmerer student a few years ago. She's currently a PhD student at UC Davis. And she was looking at feeding on phytoplankton specifically in the Cache Slough complex, which are the fresher waters in the delta of the estuary. And she found that there was a surprising high sequence abundance of cyanobacteria in the diets of this species of copepod. Cyanobacteria are considered poor food, so that was surprising. And a low sequence abundance of cryptophytes, which are thought of as good food, and they tended, at least sequence-wise, to be abundant in the water. So that triggered some additional questions. One of them was, is actual ingestion of cyanobacteria high, or is this a sequencing artifact? We have a student, Cheryl Patel, currently at SFSU, who I helped design a qPCR assay to measure ingestion of cyanobacteria. So stay tuned for that in the near future. We'll see. So moving forward, there were two recent studies. I'll talk mainly about one of them, the preliminary results from one of them, where we're investigating what actually provides food for our copepods. One of them we lovingly called Hunger Games, which I helped design this lovely logo for, where, oh yeah, the first one is Hunger Games. The second one is called the Sacramento River Nutrient Change Study, AKA we call it SUNS Baseline, SOB, shown here in respectively orange and purple. And for both of these, we collected copepods from the field, preserved them immediately in 95% non-denatured ethanol, and also collected water samples to compare what's in the diets of the copepods to the prey. And to look at the diets, I did dietary DNA analysis, sequencing 18S, region of the 18S and 16S genes. We also took other measurements of growth, reproduction, and feeding in these studies, but I'm not going to talk about those today. To back up only slightly, I mentioned the DNA-based technique before called DNA barcoding. Just to give a little more background on that, DNA barcoding is really great and important because it is the basis for the genetic database that we have, which mainly is the NCBI GenBank public repository of DNA sequences. And a common DNA barcode that we use, which is a gene region, is the mitochondrial cytochrome oxidase subunit 1 gene. And within invertebrates and many other organisms, this gene has been shown to be very good for getting you a species level idea of an organism just based on the rate of mutation of the DNA base pairs, where you can use it to identify individuals of the same species and distinguish between different species. Now, of course, it's a little fuzzy and it differs between very different phyla of organisms, but we know for zooplankton this works very well. DNA metabarcoding, again, defined earlier, is using it on a bigger scale for whole communities, whole assemblages of organisms. So for the procedure handling my copepods in the water samples, for the copepods, I cleaned and extracted DNA from sets of 5 of Pseudodiaphanous forbici. And for the water samples, filtered the water onto 0.2 micron Steravex filters. And then for library prep, I targeted the 18SV7 region and 16S gene targets. Sequenced them with aluminum-ice barcoding. And for this study, classified them using the AnaCappa toolkit primarily, especially for the 18S. We had some challenges with the 16S using generating a custom reference database for the 16S because there's too much information mainly. So we used the Silva database for the 16S and let's see. So yeah, the benefits of the AnaCappa toolkit are that you can create a custom reference database based on whatever primer set that you used for amplifying your libraries and generating the DNA sequences. And that potentially results in more accurate comparison of your unknown sequences that you get out the other end to this reference database and known species. And so I got good results with this, and you'll see downstream that yeah, it seemed to work very well. But it was sort of a test of the AnaCappa toolkit, and this actually I got off the sequencer in December, so it's only been 3 months. Anyway, so for a broad measure of sequence diversity across both studies, here I'm showing PCOAs of the assemblages in each sample, the sequence diversity in each sample, and how it compares with break-curve dissimilarities for the 18S on the left, which is eukaryotic diversity, and the 16S on the right, which is prokaryotic diversity, mostly bacteria. And for both genes, for all the samples in both studies, we saw a similar pattern in the diversity where the water assemblages were clearly more similar to each other than to the diet samples. And the diet samples in red and black spread out quite a lot more, and mostly depending on sort of where and the conditions that they were collected in, which was sort of expected. When looking at the actual diversity in these samples, first I'll show the 18S eukaryotic prey. On the left are the field copepod samples colored by a class level of ID. And I've sort of highlighted over here the ones that could be found across a majority of samples within a category. So the things that were more abundant in the copepod diets, and this is only the son of baseline study, just to sort of narrow it down, because there's a lot to look at in general. Then the son of baseline study in the copepod diets, I found that there were 2 types of green algae that were common across the samples, chlorophytes and Zygnemophyceae, possibly a sponge, a freshwater sponge, and ciliates were quite common, which is good and expected. And the water samples, there were diatoms of Cosinodiscophyceae, cryptophytes, and ciliates, and the group Spirotrichia. Maybe some bivalve DNA showed up in there from the water samples. I guess then in that case, probably eDNA, and diverse others at lower relative abundance. In the same study for the prokaryotic bacterial prey, in both types of samples, there was actually Bacteroidea, which is a common microbiome organism. So it's a little bit of a surprise to see that in both copepods in water. And alpha proteobacteria and cyanobacteria, which are common water microbes. In the copepod diet, there was a group of bacilli, which are extremophiles and spore forming, some gamma proteobacteria, which are likely a microbiome signal, and some unclassified bacteria. In the water itself, there were acidomicrobia and actinobacteria, which are known soil-dwelling microbes. So that's not surprising given how turbid and turbid the waters tend to be in these sort of tidally driven waters of the estuary. And a group of Armatimonidia, which is an aquatic plant associated microbe. So that's also not super surprising, but interesting to see. Across both studies, including the Hunger Games study, we did see cyanobacteria in both the diets and the water, and these other groups of alpha proteobacteria and Bacteroidea and gamma proteobacteria. Some of those are likely a microbiome signal in the diet, some other are actual diet items, so I still have to sort of dig into what's going on there and trying to tease those apart. And then in the 18S eukaryote assemblage, there was much more variable across both studies, but the most common and abundant in the diets were chlorophytes, and in the water were diatoms and cryptophytes. And I sort of highlighted the cyanobacteria and cryptophytes because those were also highlighted in Anne Holmes' study. So I look forward to sort of comparing and contrasting results from her work. And I'm wondering if these results hint that the eukaryotic assemblage may be more sensitive to regional variations in conditions or to predator assemblages than the prokaryotic assemblage. But it may also be the resolution of how I'm looking at these from a class level versus something more specific. I also found some interesting evidence of pathogens and parasites in many of the copepod samples. So I'm hoping to look at this with sort of an eye for health of the organisms as well. So moving to larger things, the fishes. As I mentioned, the fishes are in decline, and part of that is from the drastic reduction in chlorophyll. There's also been changes in fish food abundance and biomass. The copepods and the mysids are the most common sources of food for planktivorous fishes and some adult, and larval stages of fishes. In the copepod assemblages, there were introductions in the 90s of Pseudodiaphanous forbizae, which is now one of the most abundant copepods, and a very small cycloplecoid called Limwithona, which is shown in pink, which also tends to be highly abundant, but it is also very small. And we found that it's not often a preferred prey item or a common prey item for most of our native fishes. And the mysid assemblage has changed from being dominated by one called Neomysis mercetus to Hyperacanthamyces longirostrans. And one of the questions that I asked myself early on is the larval prey community critical for the survival of these species? If we've had such a change in what prey are actually there, we know that the larvae require easy prey in early development. Now, this is an example with cod larvae, but this holds for many fish species, that they require easy prey in early development, often copepod nauplii, and they often have a shifting gut content over development as they become more and more capable of capturing more complex and hard to capture prey. So it's quite possible that the shifts in assemblages are impacting our fish species. So that inspired my study looking at whether there are prey and larval fish diets that can help us understand the decline in fish species. And I chose to look at longfin smelt, a native threatened species in our estuary with a restricted distribution from the San Francisco estuary to Alaska. That is anadromous. It comes into the estuary to spawn, and then as juveniles, they go back out to sea every year, at least most of the population. And then the Pacific herring, which is widespread and industrial fisheries target, mainly marine, but they do also spawn in the estuary. And they also importantly have overlapping spawning timing. So when longfin smelt are present, so are Pacific herring larvae of the same sort of size and stage. What was known about the prey based on morphological ID before this study? For longfin smelt larvae, copepods were known to be the dominant prey, and there's an ongoing study also characterizing this at a higher level by master's student Jillian Burns. For juveniles, which I won't present much about the juveniles today because I haven't published it yet, but I did also look at juvenile diets, dietary tuning. Mysids tend to dominate the prey, so there's a switch from copepods dominating the prey to mysids, and that's pretty well known. In the Pacific herring, which I already highlighted that, they consume a range of plankton prey, and This is a larger list that may reflect that they're better studied because they're industrial fisheries targets, but things from rotifers to diatoms and copepods, ostracods, all sorts of plankton tend to be prey for Pacific herring. So with morphological diet analysis, of course, larval fishes are very small. They're on the order of 10 millimeters, in many cases 10 to 20, depending on the species. You have to micro dissect them under a microscope. When you open up the stomach, you hope to get something that you can identify to species, hopefully, but often you get chewed up, masticated legs of copepods or nauplii that there's no way you can identify. So there are challenges in morphological ideas of the species. So that's, of course, a clear advantage of doing dietary DNA. As far as collection goes, we did paired larval fish and zooplankton for the larval study and juvenile fish trawling with the 2 millimeter net mysid collection and zooplankton, preserved them in 95% ethanol, sorted and identified the larvae, sterilely micro dissected fish stomachs and extracted the DNA from whole stomachs, and then the DNA metabarcoding on the diets and prey assemblages with the mitochondrial CO1 gene, which I'll show here. Now, because the mitochondrial CO1 gene would also amplify fish DNA, I had to design a blocking primer to reduce amplification of the fish DNA. And that's all described in my publication that came out last year. And more details can be found there. But generally, dietary DNA mitobarcoding of the guts, just mentioned, and compared to the zooplankton assemblages, where I took a quantitative subsample and extracted bulk community DNA from that, amplified the mitochondrial C1 gene, and followed a slightly different, more basic analysis pipeline using NCBI BLAST combined with Midori, a Midori database of metazoans with the RDB classifier, and the remaining less than 80% confidence went into the unknown category. Because we noticed right away that there were a lot of unknowns and we knew that there should be cyclopoids of certain types in there, I had a NSFRU fellow work to individually barcode to identify unknown cyclopoids in our practicoid DNA and fill some gaps in my little database. And she was able to get positive confirmations on 4 common species. One of the important ones is Jungbluth tetraspina, which tends to be super abundant. So that was a win for individual barcoding. So that's just a shout out that we need to continue doing more individual DNA barcoding and make that available publicly for people to be able to categorize this unknown DNA. So what did I find? So this is a summary of how the DNA changes over development, how the prey changes over the development for longfin smelt. So there's a little bit of data from Jungbluth 2021 and some unpublished data on juveniles. I like this plot because it sort of shows that there's a switch in here from the copepod dominated prey to the mysid and amphipod dominated prey. And the less than 20 millimeter fishes are native and introduced copepods and other fishes, and mysids, amphipods, and this big unidentified arthropod group in the juvenile fishes, which is probably a type of mysid that just hasn't been barcoded yet. So I got to dig in that one a little deeper. With the herring DNA, it seemed that they have a higher reliance on cyclopoid copepods, and they consumed more Limnoidona tetraspina that were very small but abundant. And there were more sort of in this other category that were sort of low abundance sequences, a diversity of low abundance sequences that kind of got pushed into the other category, which suggests that there might be more of a generalist feeding behavior for the herring larvae. And so in general, we found, dominated by crustaceans, zooplankton. The results suggested that herring had a more flexible diet than our longfin smelt, and the prey in some cases hint at life history characteristics. For example, there was a greater amount of fish DNA in the longfin smelt than there were in the herring. Now we know with morphological diet analysis, they see fish eggs in the guts pretty often, and so we know that they eat fish eggs. This tells us what fish that they're eating. And also herring DNA was found in the lungfin smelt guts, which could hint at where they're feeding in the water column if they're eating the eggs off the substrates that the herring are laying their eggs on. So that's cool. It could also hint at schooling behavior. If they're schooling with other fish species, it's likely that the DNA of other fish species might end up entering their gut. There were many previously unknown and soft prey identified, and there were a lot of unknown and unidentified species DNA. So just need to do more DNA barcoding efforts and build better databases. It also hinted at regional or habitat-based differences in the diets. In this tidally driven system, it raised the questions of how much spatial variability is there in the fish diets. You can see I sort of highlighted more cyclopoids of a certain group in Suisun Bay, which is sort of further into the estuary, and more cladocerans in this one sample that weren't found in the San Pablo Bay. So there's sort of differences in diets in both species. And that sort of inspired a new project to sequence the diversity in the microbes to zooplankton living and restoring wetlands. So we just started field sampling this January to identify indicators of the status of wetland restoration and of the benefits being provided to higher trophic levels. With the driving questions, do wetlands in different stages of restoration provide different food web resources to the fishes that utilize them? And is there a succession in the communities of microbes to macrobes over time that can be used to indicate the progress of restoration that can be monitored by taking a water sample? So the general idea here will be multiple gene sequencing early, intermediate, and mature wetlands, collecting water samples, zooplankton and larval fish, to look at what the fish are eating in the different wetlands and how that connects to what's being provided by the wetland itself, targeting different gene regions to look at different components of the assemblage. And my grad student, Eric Ortiz, is focusing on that aspects of that for his master's thesis. Also have a new collaboration with Dr. Crow, which you may or may not have heard about if she's spoken yet. She's your visiting scientist this year, chasing manta ray environmental DNA in Hawaii using plankton. So I'm excited to go back to my roots with that project in the near future. All right, with that, final thoughts. In the age of biology, molecular tools are enabling us to explore and solve problems in novel ways. Genetic tools are useful for a wide range of aquatic studies, for DNA barcodes to ID species, to look at identification abundance of difficult to ID early life stages, blocking predator DNA and diet studies, looking at environmental DNA of the large and small, and doing dietary DNA analysis for a finer level of ID for organisms. And I'm looking forward to bringing all of this work together with interdisciplinary collaborators to solve big problems facing our ecosystems and species. Thank you very much for your attention. I know it's kind of long, and I look forward to your questions. Can't hear anything if anybody's saying anything. I see clap. Thanks, Karen. Anyway, I can't hear anything. Is it me? I don't think it's me.
Okay, can you hear us now?
Oh, there we go. Hi.
Okay, all right,
Yes.
we'll give you another round
Fabulous.
of applause.
Thank you.
Thank you. Okay, so we have the easiest way for you to ask a question in the audience is to come up here so that the microphone can pick you up. So are there any questions in the audience at the moment? There might be one on the Zoom, which can get us started. So this question is from Amber. She says, great talk. Do you have an estimate of what percentage of known copepod species have been coded?
Oh gosh, globally. Oh, actually, I found a really interesting resource at the Ocean Science meeting recently. Somebody from NOAA presented. They have a database that they've been developing called Copepodite. I think it's called Copepodite or Copepodia. I don't know. They've got a Nauplius, Copepodia, Copepodite. And one of them will show you from the known described copepod species what proportion of that has described DNA barcodes. And I think a wild estimate is maybe 10%, on the order of 10%, we'll say, have been barcoded. It's not as many as one would hope. Part of that is because the most well-studied are the planktonic copepods, but there's all sorts of groups that are parasitic or benthic or live only in sand habitats, and they're really hard to study. And there have even been copepods found in the epiphytes of trees, like redwoods. I think I read a study about copepods being found up there somehow. So they're everywhere. It's crazy, and yeah, we're just sort of chipping the surface. It could be totally off in that number, but I think it's somewhere around the less than 50 for sure.
Great. Yeah, Amber says thank you. Super interesting. So yeah, to kind of follow up on that, how much diversity do you think that DNA will uncover within copepods that has been kind of cryptic?
Cryptic species. Well, let's see. We already know that the sort of universal DNA barcodes that we use commonly still don't work super well for some groups. Like even cycloploid copepods can be hard to get with the traditional barcode, the traditional like Fulmer region CO1 barcodes. So when we really want to get those, we have to sort of work harder and try different primers and try different techniques to sort of get the information. So yeah, I don't know. It's hard to say.
Much work to be done. That was Justin's question came in right as I
Mhm.
was asking that. Has barcoding revealed any cryptic copepod species or species?
Oh yeah,
complexes?
lots of them. Yes, that's some of the work that my PhD advisor Eric Gertza does work looking for sort of cryptic species and things that are differences in something defined as one species but lives in the global ocean, but then you see these levels of difference that suggest it might be a different species sometimes. But yeah, all sorts of things. Actually Eurytemera, formerly Affinis, actually is genetically most related to one that was described a few years ago as Eurytemera caroleae in the San Francisco estuary. So that's what I've been referring to it as, because that's what I sort of found with my high throughput sequencing work. I think that was a fun one. So and that is Eurytemera is found sort of from fresh waters and some coastal waters that sort of tend to be flexible in the Great Lakes, up north in like Washington and then along the East Coast. So it's kind of widespread, Eurytemera affinis, but then they found this cryptic species caroleae that happens to be in our estuary.
Interesting.
Mm-hmm.
So do we have any questions from the audience? Anyone want to come? Yeah, come on up. Hi, Michelle. That was wonderful. Thank you. I was curious about something you said about the nutritional value of copepods as food items for other fish. And I was wondering how their diets, their phytoplankton that they consume might influence their nutritional value. Does that vary within species depending on their diet?
Oh, yes, it's complicated, but there are certain prey, like certain types of ciliates and dinoflagellates that have been described as trophic upgraders that when zooplankton eat them, they get more nutrients from the ciliate or dinoflagellate than they would from eating the things that the ciliate and dinoflagellate are consuming. So yeah, I think the prey assemblages for the zooplankton themselves do matter. And yeah, certain species, like especially in more polar regions, there are copepod species that have lipid reserves that they build up so that they can overwinter in diapause at depth. And they basically go into hibernation mode on their lipid reserves. But those copepods are really tasty food morsels for fishes and for larval fishes. So, and we know that when they don't have those lipid reserves, the fish populations don't do as well. So yeah, there are definite connections between the nutritional content of the zooplankton themselves and higher levels.
Chocolate chip cookie.
Thank you.
All right, are there any other questions from the audience? I have a question about the gut barcoding stuff from the fish, the larval fish. That was so cool. And I wonder if there are ways, or if you've thought about doing kind of paired studies where you compare what's in the water column with a similar barcoding approach versus what's in the guts of the fish to say if there's any selectivity in their feeding based on what's available.
I did not describe that well. That is one thing that was included in the study. It's hard to do the selectivity with DNA because of the potential biases of primers, and it's not quite quantitative as far as like estimating absolute abundance of biomass. But the relative quantity can be compared. And in the pub that I mentioned, the Jungbluth 2021 in environmental DNA, where I talk about the larval fish diets, I did something similar that gets an idea of selectivity using something that's used in transcriptomics to look at sort of relative expression or relative DNA comparisons of from the assemblage in the water that was also found in the diet. Were there higher or lower levels in the guts, basically? And there was a little bit of hint at, it wasn't very strong, but there were hints at selection for and against different types of prey. For example, Limnoithona tetraspina was one that was more abundant in the water column and not consumed by longfin smelt. Very odd, like there was one or two fishes, I think, that ended up with a little bit of DNA in their gut from that species. So it was selecting against the Limnoithona generally, or just unable to consume it for whatever reason. Maybe they can escape faster. There's also behavior at play, that sort of thing. So, yeah.
Yeah, neat. So we have a question from Karen. She says she's unable to unmute herself. Is it possible to unmute her? Or should I just read her question? Okay, Karen, are you there? Yeah, you're unmuted. Cool.
Hi, I think I'm unmuted. Yay. Hi.
Hey.
Hi, Michelle. Thank you.
Hey,
Great
Karen.
talk.
Thanks.
Yeah, I just wanted to chat with you a little bit. I'm super interested in the quantitative nature of eDNA. And I think I was really interested in your result where you found a really tight correlation between the number of individuals that you painstakingly counted and the number, I presume, of reads in your ddNA study. Is that right?
Let's see. The tight correlations, those were with the copepod nuclei, not dietary DNA. So that was before I got into the dietary DNA.
That was like in the
stuff.
water, I guess.
Yeah, so it was the sort of more community DNA, taking like a zooplankton sample, grinding it up, and then using, calibration of comparing the DNA copies compared to how many copies an individual has to back out the number of individuals or the biomass. Yeah, so it can be quantitative with the community DNA. With environmental DNA, it's a little trickier to get quantitative information, like the true free DNA, like with your manta rays. But I know, you probably could describe maybe more about those.
I was just
experiments.
wondering what's in your, like, you've done a lot more with this than I have. Do you think that CT is a reliable indicator of relative abundance, or like compared to number of reads in like some kind of a high throughput scenario, or like, are they off by an order of magnitude? I know it's a more, a much rougher estimate. And again, I know you can't do like absolute abundance, but just in terms of relative abundance, in your experience, what do you think about CT value as an output?
Well, I think if you're referring to the manta ray and environmental DNA type of studies,
Something like that.
Yeah, or like tracking fish populations just by the DNA floating in the water. I think that, I think using CT to detect whether there was a stronger signal or more of those potentially. Actually, there was an interesting talk earlier today that I only caught the tail end of that suggested that it's confounded a little bit if there are things like spawning events in the area. There's a lot more free DNA floating around in the water just from spermatophores and stuff from that species. I think that was an example with shrimp. So those are sort of things you have to consider as well. What are other potential sources? It could either be one big animal or a lot of small animals, or I don't know, things like that can sort of, also residence time with the water. Is there a lot of flow going on that would waft it away quickly? And if you get a high signal, that means, you know, you have a confident measure of it being there recently, or is there a high residence time and potentially that animal was there a week ago? So it's kind of, yeah, so there are a lot of complex factors, but I think in general, the CT can give you, is generally quantitative. Yeah.
Right. Yeah, it's super interesting. We're doing this study with leopard sharks, and I do think that they are using like the area where we sample as a nursery ground. So I'm sure there's like blood and they give birth to live young. So there's all kinds of stuff in the water. But do you think that you would have the same biases with the number of reads, which is very quantitative
compared to like a CT?
A CT, which is just like a doubling, right? I mean, so I mean, I guess you could mathematically say it's like off by an order of magnitude, right?
Well, I mean, it's technically kind of, it's a, with qPCR and the targeted stuff, you're measuring just one thing, you know, with the general primers, it's all of the things, but when you look at just the read count, it's sort of similar to a CT value in a way. It's just, you're looking at one versus a whole community. So
I guess counting efficiency is as in all, like, you know, RNA, seq, eDNA, seq, whatever it is, becomes a factor. So I guess each data set, I'm just like trying to think out loud with you about comparing these two types of data.
Yeah.
I guess counting efficiency is an issue with high throughp number of reads.
Yeah.
Whereas CT
and
kind of a
sequencing
gross
depth.
estimate.
Yeah, there's sequencing depth issues. There's a big push for including positive controls in mixed samples so that you can make things more quantitative and comparable from sample to sample and from run to run. Picking your positive control is a little tricky, but yeah, I think that's generally recommended. So like a known spiked amount of DNA copies from something, you know, that you know of will help you get a better quantitative measure with high throughput sequencing. Yeah.
Right. Awesome. Thank you.
Nice. Well, thank you so much again, Michelle, for your talk today. And I think that we are, no more questions. Yes, I think that we're good, and we're good in the chat. So we'll just give you another round of applause, and thank you for your talk.
Thanks, everyone. And feel free, if you have any follow-up questions, reach out to me. My email is pretty easy to find.
Nice to meet you, and hope to welcome
nice
you in
meeting you.
person at some point.
Yeah, thank you.
All right, thanks everyone.
Bye.
Bye.